So dear students, continuing with the development, that is embryology classes. Previously we have seen the development during first week, second week, third week. So if you just go with little revision, the phases of embryonic development we saw, like first phase is growth, where involves the cell division, and second phase is the morphogenesis, where we learnt about the gastrulation. And the second phase in morphogenesis, it also includes the development of somites and foldings of embryo. In the third phase is the differentiation. So these are the three phases which are associated with the embryonic development. So today we are going with the development of 4th to 8th week. So by this we will complete most of the general embryology. So by the end of the third week, the primitive node, which we know it differentiates into 3 germ layers, that is ectoderm, endoderm, mesoderm. And mesoderm, intraembryonic mesoderm, it grows vastly except at the procaudal plate. And posteriorly cloacal membrane. So this is the superior view of embryo, where I marked procaudal plate and cloacal membrane, and we can see the central axis having neural tube. So this is the neural tube in cross section, and below the neural tube, so beneath it, there is a notochord. So notochord can't be seen from superior view. In cross section we can appreciate notochord. So on each side of the notochord and neural tube, this intraembryonic mesoderm, it is further differentiated into 3 longitudinal columns. The 3 longitudinal columns are paraxial mesoderm, which is present on each side of the neural tube. So this is para, means on each side, axial on each side of the axis. So here is the paraxial mesoderm, which is present on each side of the developed neural tube. And further laterally, this intraembryonic mesoderm we can differentiate into intermediate and lateral plate mesoderm. So this is intermediate mesoderm, and further laterally we have got lateral mesoderm, that is on still more lateral side. So this is paraxial, this is intermediate, this is lateral plate mesoderm present on each side. Paraxial mesoderm consists of the longitudinal columns which are further differentiated into segments. So the intermediate mesoderm, we know it is sandwiched and it forms urinary tract and genital tract in adults. And lateral plate mesoderm, it is continuous with the extra embryonic mesoderm at the covering of the yolk sac and amnion, where we have got extra embryonic mesoderm. So this lateral plate, it is continuous with extra embryonic mesoderm. So each lateral plate mesoderm, it splits horizontally by dorsal somatic parietal mesoderm, which underlines the ectoderm, and ventral splanchnic mesoderm, that is called visceral mesoderm. So here is the cross-sectional view of the embryo. The blue lining is the amniotic cavity, and cavity with yellow lining is the yolk sac cavity. And here we have got paraxial mesoderm, which forms somites. This is the paraxial mesoderm which forms somites, and here is the intermediate mesoderm. So here is the coelom present within the mesoderm, and it splits into, the lateral plate is further splits into dorsal somatic mesoderm, that is parietal mesoderm. Here is the dorsal one, somatic mesoderm or parietal mesoderm. Parietal means body wall, so it is associated with the body walls. Whereas the ventral, which continues along the yolk sac, this is ventral, which forms visceral mesoderm. So that is about this lateral plate mesoderm. Visceral is otherwise called as splanchnic visceral mesoderm, which overlies the endoderm. So it is continuous here. We can see on the other side also it is continuous along the endoderm here. Whereas the parietal it goes along with the amniotic sac onto the dorsal side. And the space between these layers, that is the body cavity. So that forms the 𝐒𝐿𝐽. So you are seeing the gap here. I have put arrow on the other side also we can see this gap. So this gap it forms 𝐒𝐿𝐽. The space between these layers becomes the body cavity, that is the 𝐒𝐿𝐽, which stretches from future neck region to the posterior part of the body. And during later development, the right and left side of the 𝐒𝐿𝐽 is fused by folds of the tissues which extends from the somatic mesoderm, dividing the 𝐒𝐿𝐽 into separate cavities, that is the thoracic cavity in the thorax region and abdominal cavity in the abdomen region. So 𝐒𝐿𝐽 is subdivided into pleural pericardial cavities which are present in the thoracic region, whereas the peritoneal cavities which are enveloped by the peritoneum, so that peritoneal cavities are observed in the abdomen region. Pleuropericardial are present in the thorax and heart, and whereas peritoneal it is associated with the abdomen. And parietal layers forms the parietal layer of the pleura, pericardium and peritoneum. Parietal layer developed from the dorsal somatic lateral plate mesoderm. So dorsal somatic lateral plate mesoderm will form the parietal layers of all these, that is the pleura, pericardium and peritoneum. Whereas the visceral layers of pleura, pericardium and peritoneum develop from the ventral visceral lateral plate mesoderm. And paraxial mesoderm, towards the end of the third week, the paraxial mesoderm, that is the caudal to the aortic placodes, that is ear placodes, caudal to the aortic placodes, this paraxial mesoderm differentiates to form a condensed pair of segments which are called as somites. So what is meant by somites? Soma means body. So these are like a segmented bodies which are present on each side of the developing neural tube. If we see these somites, the first pair of somites, they appear in the future occipital region of the embryo. That is, they appear first in the cranial region. Occipital region means cranial head region. And further they start growing caudally. So craniocaudally the segment starts forming. Each segment is called as somatomere. So each segment, usually around 38 pairs of somites are formed during 20th day to 30th day of gestation. So approximately 38 pairs of somites are formed during 20th to 30th day of gestation or intrauterine life. So this pair of somites, that appearance, it is very particular. So by this, clinician, they can guess the age of the embryo by seeing the number of somites which are formed. So by the end of the 5th week, if we observe, around 42 to 44 pairs of somites are present. So by the end of the 5th week, around 42 to 44 pairs of somites are present. So somites form a distinct surface elevation over the embryo. Now these somites form a prominent feature between 4th to 5th weeks of gestation. So during 4th to 5th week of gestation, the somites are more prominent, and the process of development of somite is sequential, and therefore it is used to stage the age of the embryo based upon number of visible pairs of somites. So by seeing the number of somites, a clinician can guess the age of the embryo. Components of somites. Somites are further differentiated into, so if we see each segment somites present on the neural tube, this is the neural tube. The somites can be further differentiated into two parts, sclerotome and dermomyotome. So it can be divided into sclerotome. The other component is dermomyotome. So sclerotome, it forms the bony component. So sclerotome derives to form the axial skeleton, that is the vertebra and ribs, and sclerotome surrounds the notochord, developing notochord, which is present beneath the neural tube. So it envelops the developing, surrounds the notochord and projects posteriorly to surrounds the neural tube also, and further it is divided into ventral sclerotome, lateral sclerotome and dorsal sclerotome. Ventral sclerotome forms the vertebral body and annulus fibrosus, which is a part of intervertebral disc. Lateral sclerotome forms the vertebral arch, that is pedicel and laminae. And dorsal sclerotome forms the spinous process of the vertebra. So these are the parts of the vertebra which are derived from each part of sclerotome. So ventral forms the ventral body of the vertebra. So if you see the vertebra, this is the body, transverse process, and posteriorly we have got the spinous process. And this is the vertebral canal. This is just a schematic picture for you to understand. So body is derived from ventral sclerotome and dorsal sclerotome will form the spinous process. Lateral sclerotome will form the vertebral arch, including laminae, that is this laminae on each side, and pedicels, that is lateral sclerotome. Next about the dermomyotome. As the name suggests, dermo means it forms dermis, myotome means muscle. So myotomes forms the skeletal muscle, which is a precursor which forms the muscles of the back and the body wall and the limbs. So all the muscles in the limbs, back of our body and body wall are all derived from this myotomes. Whereas the dermatome, which forms the dermis of the back. So dermis of the back in the dorsal region is derived from dermatome. Now let's see about the foldings of embryo. Now we know the mesodome is formed, and we saw the differentiations of the mesodome during 4th and 5th week. So along with the somites development, there will be a folding of embryo. So initially if you see the embryo, it is a flat trilaminar germ disc. So this is a flat trilaminar germ disc containing the layers, and we know that the outermost is the trophoblast here, and a splanchnopluric mesodome which is along the yolk sac, and this is the connecting stalk, and here is the allantois. So this is how initially we learned till the 3rd week of development. So later in 4th week and 5th week we see the folding of the embryo, where the trilaminar germ disc, which is like a cylindrical embryo, it folds in such a way that there is both median foldings and horizontal foldings. So here is the sagittal view of the embryo showing the median foldings. The median foldings are head fold and tail fold. So this is the head side, cranial side, head fold. The other side is the tail fold of embryo. And we can label here, this is the amniotic cavity, and here is the developing embryo here. And because of this head and tail fold, what happens? The yolk sac partly get absorbed into the developing embryo, further divided into foregut, the midgut, which connects to the stalk of the yolk sac, which is called vitellointestinal duct. This is the midgut, and this is the hindgut of embryo. And from hindgut we have got a projection which is called as allantois. And the stalk-like connection which is between the midgut and the yolk sac present outside, this connection is called as vitellointestinal duct. This whole thing is considered as connecting stalk. So this is how the head and tail fold occurs. So let us discuss little more in detail. So fold in the median plane, there are head fold and tail fold. Talking more about this head fold, at the beginning of 4th week, so the first fold which forms is the head fold, not the tail one. So in the beginning of the 4th week, the cranial part of the neural tube develops into a primitive brain. So if you see this neural tube, which is formed here. So this is the bulging which is formed within the head fold. So because of the eventual growth of this forebrain, which is present within the head fold of the embryo, so the forebrain grows beyond the oropharyngeal membrane and developing into the head fold. Gradually, what happens? The developing heart, which is overhanging below. So now we see here, this is discoid-like cylindrical embryo, flat, developing trilaminar germ disc. Here we can see the growth of brain where the amniotic cavity projects ventrally, cranially, and we can see here this is the developing heart where we can see the pericardial cavity and heart tube, and this is septum transversum which is growing caudal to the pericardial cavity, and here is the yolk sac. This is allantoic diverticulum, cloacal membrane, and this is little part of primitive stick which is still remnant, and the red line is the notochord, and this is the neural tube. This yellow one is the neural plate which forms into neural tube. So if you see here still more closely, so this is the head fold. The pericardial cavity, initially it was cranial, it becomes ventral. And here is the septum transversum which forms the diaphragm mainly, and allantoic diverticulum present in the hindgut. This is foregut, this is midgut, and this is hindgut. And we can see this is vitellointestinal duct. Okay, and here is the cloacal membrane and primitive streak, and we can see still notochord is persistent here, and this is the neural plate which forms neural tube, and this is the buccopharyngeal membrane. So we can see the head fold grows beyond the buccopharyngeal membrane or procaudal plate. During the development, the head fold, the part of the endoderm of the yolk sac, that is the umbilical vesicle, which is incorporated within the head fold, forms the foregut, and the part of the yolk sac which is incorporated in the tail fold is called hindgut. In between, the communication with the vitellointestinal duct which communicates with the yolk sac externally, that is forms later into midgut. So foregut is the precursor of pharynx, larynx. So foregut in later it forms pharynx, where we see development of pharyngeal arches. It forms larynx. It forms esophagus, respiratory tract, and it also forms the stomach and initial part of duodenum. So all these are derived from the foregut. So what are the effects of having this head fold? So after the folding of the head, the septum transversum lies caudal to the heart. So initially if you see here previously, so the septum transversum is here which lies caudal to the heart, forming like a partition, and which turns to form the fibrous pericardium, that is a part of pericardium, and it also forms the central tendon of the diaphragm, and the rest of it invades into hepatic diverticulum to form the connective tissue of the liver, and it gives rise to the ventral mesentery of the foregut. So thus the foregut is located between the developing brain and the heart. So between the developing brain and the heart, so this part is the foregut. So cranial end of the foregut separated from the stomatodium by a membrane called as buccopharyngeal membrane, which is also called as oropharyngeal membrane. Before folding, the coelom consists of flattened horseshoe-shaped cavity. After folding, the pericardial coelom lies ventral to the heart, cranial to the septum transversum. Within the head fold, anterior above to the septum transversum, it is present. And embryonic coelom divides into intraembryonic and extraembryonic coelom because of this head fold. And intraembryonic coelom forms the coelomic cavity which is absorbed inside the embryo during the head fold. So during the formation of the head fold, these are the things. And let's talk about the tail fold. So the distal part of the neural tube, that is the spinal cord, grows forming the tail fold. So during the folding of the part of the yolk sac is incorporated into the tail fold, forming the hindgut. So this part HHG which I represent, it forms the hindgut. And the distal part of the hindgut is separated from the proctidium, that is the future anal opening, by a cloacal membrane. So this is the cloacal membrane. So here is the cloacal membrane, which is a bilaminar membrane. It is separated from the hindgut by means of proctidium. So stomatodium, future mouth. Proctidium, future anal canal. So effects of the tail fold, what happens? The primitive streak which lies cranial to the cloacal membrane. So you can see here it is cranial to the cloacal membrane. It becomes caudal. So because of the folding. So cranial to the cloacal before folding. After folding, it lies caudal to the cloacal membrane. The connecting stalk, that is the primitive umbilical stalk cord, it is attached to the ventral surface. So you can see the connecting stalk, it is attached to the ventral surface. You can see the connecting stalk which is attached to the ventral surface after forming the tail fold. And the folding of the embryo in horizontal plate, there are two folds which are called as right and left folds. So what all we saw is the head fold and tail fold which is seen in the median plane, that is in the sagittal section. We can appreciate this head fold and tail fold. If you take a transverse section along the embryo, then we can see the two lateral folds. So if you take here the section, we can see the fold here, the coelomic cavity which is extending into this, forming the two lateral folds. You can see the projection of the amniotic cavity projecting on each side. So initially this projects on each side to form the two lateral folds. So the folds are right and left lateral folds. These lateral folds are produced because of rapidly developing somites and spinal cord. And the lateral folds form due to the folding of ventrolateral walls on each side. So as the folds form in all four directions, the part of the endoderm of the yolk sac layer is incorporated into the embryo as the midgut, which we saw earlier. And at the beginning of the folding, the midgut widely communicates with the yolk sac. So you can see this. First, there is no midgut at all in the first picture. And second, we can see the part of this forms the midgut. And you can appreciate its communication from here to here with the yolk sac cavity. As the foldings form, the midgut initially it has a wide communication with the yolk sac. Later, because of these folds, the communication gradually constricts to form a vitello-intestinal duct. So this is the vitello-intestinal duct. As the umbilical cord forms the connecting stalk, which shifts ventrally and due to the fusion of the lateral folds ventrally, and the communication between the intraembryonic and extraembryonic cavities also reduces because of formation narrowing this connecting stalk. And amniotic cavity expands all around the developing embryo. So you can see this blue one is the amniotic cavity which surrounds all around the developing embryo, and eventually it obliterates the extraembryonic coelom. The amnion forms the epithelial covering of the umbilical cord. Further, this amnion continues with the epithelial covering of the umbilical cord. So it forms a covering over the umbilical cord. So this completes the development up till the 5th week of embryo. And next we shall move on to the 6th week and gradually 7th and 8th weeks. So we know now folding of the embryo is formed. So here it is anterior view of the embryo. Initially we can see it is discoid like, having buccopharyngeal membrane, cloacal membrane caudally, and we can see the head bulge forming. and stomatodium which forms the future mouth, and pericardial bulge which contains the heart tube, and this is the cloacal opening. This is the umbilical opening which is formed here, narrowing. In sagittal section we can see as head bulge. This is the stomatodium which forms the future mouth, pericardial bulge, and the umbilical opening. This is the tail fold. So this is how we learned till the 5th week of development, and we can see the segments, somites are also formed, and we can see now having embryo, head fold, tail fold. Okay, and this is the pericardial bulge, and this is the stomatodium which forms the future mouth covered by buccopharyngeal membrane, and we can see the umbilical cord, and we can also see upper limb buds which I am going to discuss now, and this is the lower limb bud. So upper limb bud, lower limb bud on. So this is the sagittal view of the embryo where we can appreciate all these things. So let's talk in little more about the 6th week of development. The milestones of the 6th week of development, the main milestone are reflexes. So in 6th week the embryo exhibits the reflex response to touch. So regional differentiation begins in the 6th week of gestation. So the upper limb bud grows as elbows and large plate of hands. We can see here, appreciate large plate of hands are observed during 6th week of development, and the hand plate shows formation of the digits, and it is termed as digital race, which like appears to be like a webbed feet. So we can see like a webbed feet in within the hand plate, and spontaneously the movements like twitching of trunk and limbs are observed, and development of the lower limbs are seen during 4th to 5th days later to the upper limb growths. So once the upper limb bud forms and it starts growing, after then 4th and 5th days, 5 days later, we see the appearance of lower limb buds. The other changes which we observe during 6th week of development is appearance of pharyngeal arches. You are seeing here 1, 2, 3, 4. 5th one will disappear, forms and disappear. 6. So we can see the appearance of pharyngeal arch. First pharyngeal cleft form the external acoustic meatus. So the cleft forms the external acoustic meatus, which we will discuss along with the pharyngeal arch. And we can see the optic placodes which differentiates to form the eyes are clearly observed. So this is optic placodes. And we also see the growth of the head. So head grows larger in relation to the trunk, and due to the head fold, the head is bent over the cardiac prominence. So we can see the pericardial bulge containing cardiac prominence. It is called cardiac prominence too. So we can see the head fold is ventral to the cardiac prominence. The intestines start growing in the extraembryonic coelom, exhibiting a physiological umbilical hernia. So these are all the things happening during 6th week of development. So talking more about this 7th week, we will go further into 7th week. So during the 7th week, the limb grows with a considerable change, and notches appear between the digital range. So each one will form, the hand plates will form into digits. So digital rays will turn to form digital digits during 7th week. And ossification of bone limbs has begun now. So during 6th week we see the first bone to start ossify is the clavicle, and later during 7th and 8th week we see the other bones. The primary center for the other bones also will appear, and communication between the primitive gut and the yolk sac constricts to form a narrow canal which is called as vitellointestinal duct. So this umbilical cord contains vitellointestinal duct, which communicates with the midgut which is growing outside the embryo. And 8th week, so moving on after 7th week, 8th week we see the ossification of lower limb bones begin in the 8th week. The scalp starts differentiating with the formation of vascular plexus. So we see the development of scalp also. So at the end of the 8th week, all the regions of the limbs are apparent now. So now we can distinguish all the 4 limbs of the embryo. And purposeful limb movements first occur during 8th week of intrauterine life. And at the end of 8th week, the embryo has distinct human characteristics with a large sized head. And the intestines still exhibit physiological hernia during 8th week. So the physiological hernia, it happens from 5th week to 10th week of intrauterine life. And sexual differences starts appearing, that is external genitalia, and which are not distinctly observed, but we can still start appearing during 8th week of intrauterine life. And the end of the 8th week, the size of the embryo reaches to the size of 5 mm. Means almost half a centimeter the size of the embryo is. And so summary of this 4th to 8th weeks of development, we learnt about differentiation of somites during development of the 4th to 5th week. And we saw the derivatives of somites today, and we also learnt the foldings of embryo, that is the head fold, tail fold, and 2 lateral folds, and development of primitive gut into foregut, midgut, hindgut, which gets incorporated inside the embryo. And we know the development of oropharyngeal membrane and heart are shifted to the ventral side of the embryo, which were like cranial end. The oropharyngeal was at the cranial end, it shifts to the ventral side. And terminal part of the hindgut, it gets dilated, which is called as cloaca, and it is separated from the tail end, that is by the cloacal membrane, which forms the distal end of the cloaca. The tail fold results by shifting the cloacal membrane and allantois and connecting stalk to the ventral side of the embryo. All these things shifts to the ventral side of the embryo. And we also know the formation of the vitellointestinal duct, which is the communication between the midgut and the yolk sac. Initially it was a very widened communication. Later it gets constricted to form the vitellointestinal duct. The 3 germ layers now differentiated into various tissues and organs by the end of embryonic period. So by the end of the 8th week, all main organs systems have been established now. The external appearance is greatly like a human during 8th week of intrauterine life, and vital structures like brain, heart, liver, somites, limbs, eyes, ears, and nose are developed by the end of the 8th week of intrauterine life, and most of the orgogenesis of external and internal structures are developed during 4th to 8th week. So 4th to 8th week is the period of orgogenesis. So these weeks are considered as the critical period of development because a minor development disturbance can cause a major birth defects. So this completes the growth of the embryo, that is the development of the embryo during 4th to 8th week of gestation. Thank you.